Hey there, tonight we're slipping quietly into a world often overlooked, yet brimming with astonishing life. We'll explore the green and hidden intelligence of plants, uncovering secrets whispered by roots and the clever strategies of leaves. It's a journey into botanical wonder, perfect for winding down. If you enjoy these quiet explorations, please consider liking this video and subscribing to the Sleepless Scientist for more calm dives into the mysteries of our world. Now, let's travel back in time to an era when the very idea of a meat-eating plant seemed like pure fiction. Imagine the disbelief, the quiet shock of early naturalists encountering something truly extraordinary. It began with whispers from the New World, tales of plants that didn't just stand still and photosynthesize, but actively ensnared. These reports challenged the very definition of what a plant could be. One of the most captivating was a curious specimen from the bogs of Carolina, the Venus flytrap. Its leaves, tipped with what looked like tiny teeth, were unlike anything seen before. The initial observations were met with skepticism. How could a plant move with such deliberate speed? It seemed to defy the fundamental laws of botany as understood at the time. Even Carl Linnaeus, the father of modern taxonomy, initially found it so perplexing that he refused to classify it as a true plant, famously calling it a miraculum naturae, a miracle of nature. The first detailed accounts described its mechanism, delicate trigger hairs, and a snap so swift it could trap a fly in an instant. This was not passive existence, this was active predation. Imagine the sheer wonder watching a plant close its jaws around an unsuspecting insect. It was a silent, green hunter, patiently waiting for its moment. This phenomenon sparked intense debate. Was it a mere mechanical reaction or something more akin to animal behavior? The scientific community grappled with this botanical enigma. Centuries later, one of the greatest scientific minds, Charles Darwin, turned his meticulous gaze towards these peculiar plants. He was captivated by their apparent intelligence. Darwin didn't just observe, he experimented. He spent years in his home laboratory, systematically testing the sensitivities and digestive powers of various carnivorous species. He offered them tiny bits of meat, egg white, and even his own hair, carefully documenting their responses. His patience was boundless, his curiosity insatiable. Darwin discovered that the flytrap was remarkably discerning. It ignored raindrops or inert objects, snapping shut only when trigger hairs were touched twice or by a moving insect. This suggested a form of memory or counting, a rudimentary processing of information that was utterly astonishing for a plant. It challenged conventional biological thought. He also confirmed their digestive abilities, observing how the traps secreted enzymes that slowly dissolved their prey, absorbing the nutrients. His comprehensive work, Insectivorous Plants, published in 1875, was a landmark. It firmly established the reality of carnivorous plants and their complex adaptations. But the Venus flytrap was just one example. Across the globe, other botanical predators had evolved equally ingenious, yet different strategies. Enter the pitcher plants, a diverse family with a completely different approach to trapping. Instead of snapping jaws, they offered a deadly, alluring invitation. These plants form deep, cup-like structures, often beautifully colored and patterned, that act as passive pitfall traps. Insects are drawn by nectar and scent. The rim of the pitcher is often incredibly slippery, especially when wet. An insect, lured by the promise of a sweet meal, loses its footing and tumbles inside. Once inside, escape is nearly impossible. The walls are often lined with downward-pointing hairs or a waxy, frictionless coating, preventing any upward climb. At the bottom of the pitcher lies a pool of digestive fluid. This isn't just rainwater, it's a concoction of enzymes, acids, and wetting agents. These fluids break down the trapped insects much like an animal's stomach. The plant then absorbs the released nutrients, a silent, slow feast. Some pitcher plants are even more specialized, with lids that prevent rain from diluting their digestive fluid, ensuring maximum efficiency. The diversity among pitcher plants is astounding, from tiny ground-hugging species to giant ones capable of trapping small vertebrates, though this is rare. But why did plants evolve such elaborate, energy-intensive traps? The answer lies in the harsh environments where they thrive. Carnivorous plants typically grow in bogs, fens, and other wetlands where the soil is profoundly nutrient-poor, especially lacking in nitrogen and phosphorus. These conditions make it difficult for most plants to obtain essential building blocks for growth. Insects, however, are rich in these very elements. Thus, predation became a clever evolutionary strategy, a way for these plants to supplement their diet and survive where others could not. It's a testament to life's adaptability, a demonstration that even the most seemingly passive organisms can develop astonishing methods for survival. Modern science continues to unravel the molecular intricacies of these traps. We now understand the genetic pathways that control their formation and function. Researchers study the precise mechanisms of their digestive enzymes and the chemical cues they use to lure their prey, revealing layers of complexity. The study of carnivorous plants has also provided insights into plant movement and signaling, showing how plants can respond to their environment with precision. These botanical predators remind us that the plant kingdom is anything but static. It is dynamic, innovative, and full of surprises. From the snapping jaws of a flytrap to the alluring depths of a pitcher, these plants are true marvels of evolution, perfectly tuned to their unique ecological niches. Their story is one of resourcefulness, turning a disadvantage into a unique advantage, transforming the very definition of plant life. The wonder of these meat-eating plants continues to captivate, a reminder of the endless ingenuity found in the natural world. Their silent hunts unfold in hidden corners, a testament to life's persistent drive to thrive, even in the most challenging conditions. As we drift into slumber, let the image of these clever leaves linger. Next, we'll delve deeper, exploring the ancient, hidden networks that connect entire forest communities. Beyond the individual marvels of carnivorous leaves, another layer of botanical wonder awaits, hidden beneath our very feet. It's a subterranean world, a silent, intricate tapestry woven through the soil. For a long time, the prevailing view of a forest was one of fierce competition. Each tree, a solitary giant, battling its neighbors for sunlight, water, and precious nutrients. Yet, long before scientific instruments could peer beneath the surface, indigenous peoples held a different understanding. They spoke of forests as interconnected families, living communities. This ancient wisdom suggested a deeper bond, a hidden language, far more complex than simple rivalry. But how could such a thing be? What could possibly link these silent giants? Scientists, too, began to observe peculiarities. Trees, even those struggling, sometimes showed unexpected resilience. Could their roots be doing more than just anchoring them? The answer, it turned out, lay not just with the trees themselves, but with an often overlooked kingdom, the fungi. Specifically, the vast, intricate networks of mycorrhizal fungi. Mycorrhizae are a symbiotic partnership, a mutual agreement between fungi and plant roots. The word itself means fungus root, a perfect descriptor for this ancient alliance. In this partnership, the fungi receive sugars, carbohydrates produced by the plant through photosynthesis. It's a sweet deal for organisms that cannot make their own food. In return, the fungi, with their incredibly fine and extensive threads called hyphae, act as an extension of the plant's root system, reaching far beyond what the plant could manage alone. These fungal threads are far more efficient at absorbing water and crucial nutrients like phosphorus and nitrogen from the soil, delivering them directly to the host plant. But the true revelation came when scientists realized these fungal networks didn't just connect one fungus to one tree. They connected many fungi to many trees, forming a vast, underground web. Early ecological studies provided hints, but it was the persistent work of researchers like Suzanne Simard in the Canadian forests that truly illuminated this wood-wide web. Simard, a forest ecologist, noticed something peculiar. When she removed a mother tree, the surrounding saplings often struggled, even if they had ample light and water. She hypothesized that the older, larger trees were somehow supporting the younger ones, not just through shade or competition, but through a direct, underground exchange. Proving this invisible connection, however, was no easy task. It required innovative thinking and meticulous experimentation in the complex environment of the forest. Simard and her team turned to isotopic tracers. These are like tiny, harmless labels attached to carbon molecules, allowing scientists to track their movement through living systems. They introduced radioactive carbon dioxide, a labeled gas, to specific trees. The trees would photosynthesize, incorporating this labeled carbon into their sugars. The question was, would this labeled carbon stay within the donor tree, or would it travel to its neighbors, even those of different species, through the fungal network? The results were astonishing. Labeled carbon, initially absorbed by a single tree, was detected in the roots and even leaves of nearby trees, including firs, birches, and cedars. This was direct evidence of nutrient sharing, a radical departure from the long-held belief that trees were solely engaged in a cutthroat battle for resources. Cooperation was happening. Further experiments using labeled nitrogen confirmed that other vital nutrients were also exchanged through these fungal superhighways, enriching the entire forest community. This led to the powerful concept of mother trees, large, old, dominant trees that served as central hubs in these networks, nurturing countless younger saplings. These mother trees could transfer carbon, nitrogen, and even water to their offspring, increasing their survival rates, especially in shaded or resource-poor conditions. This discovery had profound implications for forestry practices. Clear-cutting, for instance, could sever these vital connections, leaving young trees vulnerable. But the wood wide web wasn't just about sharing food. It also served as a communication network, a botanical internet where trees could talk to each other. When a tree was attacked by insects or infected by disease, it could send out chemical distress signals through the fungal network. Its neighbors would then receive these warnings. Upon receiving such a signal, healthy trees could increase their defenses, producing protective chemicals or strengthening their cell walls, preparing for a potential threat. This challenged the long-held view that plant interactions were primarily competitive or through airborne volatile compounds. The underground web was a far more direct, intimate connection. Imagine the sheer scale. A single teaspoon of forest soil can contain miles of fungal hyphae. Multiply that by an entire forest and the network becomes mind-boggling. This partnership isn't new, it's ancient. Fossil evidence suggests that mycorrhizal fungi were crucial for plants to colonize land hundreds of millions of years ago. Without this fungal assistance, the very first plants might never have thrived on rocky, nutrient-poor land, underscoring the fundamental importance of this relationship. Today, advanced DNA sequencing allows researchers to identify the diverse fungal species within these networks and map their connections with unprecedented detail. They are discovering that not all fungal species connect to all trees equally. There's a subtle specificity, a preferred pairing that shapes the network's architecture. The wood wide web is not static. It shifts and evolves with seasons, with changing environmental conditions, and with the growth and death of its members. Understanding these subterranean connections is vital for preserving forest health and biodiversity, especially in the face of climate change and habitat fragmentation. The wood wide web paints a picture of forests not as collections of individuals, but as complex, intelligent superorganisms where every member plays a role in the collective well-being. It's a silent, constant hum of cooperation, a hidden testament to nature's profound capacity for partnership, challenging our human-centric notions of intelligence and interaction. This deeper understanding reminds us that much of life's most intricate work happens out of sight, demanding patience and a willingness to look beyond the obvious. The forest floor, once just dirt and roots, now reveals itself as a bustling metropolis of exchange and communication, a marvel of biological engineering. As these hidden networks continue their quiet work beneath the trees, we are left to ponder the subtle whispers they carry. Next, we will explore the even more intimate, secret language spoken between individual roots, a conversation of chemistry and subtle signals. Our journey beneath the forest floor revealed the vast, ancient connections of the wood wide web. But even more intimate conversations occur, not between entire communities but between individual roots, a silent dialogue of chemistry and subtle signals. For centuries, the growth of roots was largely a mystery. They seemed to simply descend, drawn by gravity, passive anchors in the soil. Yet, observations hinted at something more deliberate, a hidden intelligence guiding their path. Early botanists noticed that roots didn't just grow straight down. They would curve, bypass obstacles, and seemingly seek out areas of moisture. This suggested an active decision-making process, albeit a slow, chemical one. The first major breakthrough came with the discovery of plant hormones, tiny chemical messengers that orchestrate growth and development. Among the most crucial were the auxins. In the late 19th century, Charles Darwin and his son Francis conducted pioneering work on plant movements, observing how shoots bent towards light. While focusing on stems, their work laid groundwork for understanding growth regulators. They hypothesized that decades later, in the 1920s, Dutch scientist Fritz went isolated this mysterious substance. He extracted it from oat coleoptiles, the protective sheaths covering young grass shoots, and named it auxin. Whence experiments showed that auxin moved from the tip of the coleoptile downwards, promoting cell elongation. More auxin on one side meant faster growth, causing the plant to bend. While auxins largely promote stem growth, their role in roots is more nuanced. In roots, higher concentrations of auxin actually inhibit elongation, a delicate balance that guides their downward path. This discovery was profound. It meant that plants weren't just passively growing, they were actively directing their development through a complex interplay of internal chemical signals. Other hormones like cytokinins, gibberellins, and abscisic acid were soon identified, each playing a crucial role in root initiation, branching, and responses to stress. Roots had an entire chemical orchestra. But the story didn't end with internal signals. Researchers began to suspect that roots weren't just responding to their own internal chemistry, but also to signals from their environment and even from other plants. Imagine a root growing through the soil. It's not just absorbing water and nutrients, it's constantly sampling its surroundings, detecting changes in moisture, mineral availability, and the presence of other living things. This led to the concept of chemical communication between roots. Plants release a diverse array of compounds into the soil known as root exudates. These are not waste products but deliberate messages. Some exudates act as attractants, drawing beneficial microbes or signaling to mycorrhizal fungi to form those vital partnerships we discussed earlier. Other chemicals can be defensive, deterring harmful nematodes or pathogens. A root, sensing a threat, can release compounds that make the surrounding soil less hospitable to invaders. Perhaps most fascinating is the evidence of roots communicating with neighboring plants. This isn't just the wood wide web, it's a more direct, localized whisper between individual roots. Experiments have shown that roots can distinguish between their own kind and the roots of other species. They might grow more aggressively towards kin or actively avoid competitors. For instance, some plants, when grown next to a genetically identical clone, will reduce their root growth, as if recognizing a family member and avoiding unnecessary competition. However, when placed next to an unrelated plant, their roots might grow more vigorously, expanding their territory to outcompete the stranger for resources. This suggests a sophisticated level of self-recognition and kin recognition occurring at the root level, mediated by specific chemical signals released into the soil. Beyond recognition, roots also demonstrate remarkable navigational abilities. They don't just passively absorb, they actively forage, exploring their environment. Researchers have used clear gel matrices to observe root growth in controlled conditions. They watch as roots extend, seemingly sniffing out nutrient-rich patches. When a root encounters a dry patch, it can redirect its growth towards areas of higher moisture. This hydrotropism is a crucial survival strategy, especially in arid environments. Similarly, roots exhibit chemotropism, growing towards beneficial nutrients like nitrates and phosphates. They detect concentration gradients and follow the scent to their source. They also demonstrate thigmotropism, responding to physical contact. A root might grow around a stone, sensing the obstacle and altering its path to bypass it without damage. These directed movements are not simple reflexes. They involve complex sensing mechanisms at the root tip, processing environmental cues and translating them into growth decisions. The root cap, a small thimble-like structure at the very end of each root, is thought to be the plant's brain for these subterranean explorations, sensing gravity, moisture, and obstacles. This intricate behavior highlights the astonishing intelligence of the botanical world. Roots are not merely conduits, they are active explorers, making choices and adapting their strategies. This brings us to the concept of the rhizosphere, the narrow zone of soil directly influenced by root secretions and activity. It's a bustling ecosystem, a constant dialogue. Within this rhizosphere, roots engage in a complex dance with countless microorganisms, bacteria, fungi, protozoa. This is where much of the plant's nutrient acquisition and defense truly happen. The plant provides the microbes with sugars and other organic compounds, fueling their growth. In return, these microbes perform vital services. Some bacteria fix atmospheric nitrogen, converting it into a form plants can use. Others help solubilize phosphorus or produce growth-promoting hormones that benefit the root. This constant exchange forms a feedback loop. The root shapes its microbial community, and in turn, that community influences the root's health and growth. Understanding these rhizospheric interactions is critical for sustainable agriculture. By fostering beneficial microbial communities, we can reduce the need for synthetic fertilizers and pesticides. The roots, far from being silent, passive structures, are constantly communicating with themselves, with their neighbors, and with the microscopic world around them. Their whispers are chemical signals, their movements deliberate responses. It's a language of survival, cooperation, and astonishing adaptability hidden just beneath the surface. This silent, ongoing dialogue shapes entire ecosystems, determining where plants grow, how they thrive, and how they respond to the ever-changing challenges of their environment. The patience required to uncover these subterranean secrets mirrors the slow, deliberate growth of the roots themselves. Every observation, every experiment adds another word to this silent language. As the roots continue their quiet work, we are left to ponder the depth of their understanding. But what happens when the signals aren't just chemical, but involve the passage of time itself? Next, we'll explore how flowers count and remember, revealing another layer of botanical brilliance. As the roots continue their quiet work, we are left to ponder the depth of their understanding. But what happens when the signals aren't just chemical, but involve the passage of time itself? Tonight, we turn our gaze upwards towards the blossoms that adorn our world. We explore how flowers count and remember, revealing another layer of botanical brilliance. For centuries, farmers and naturalists observed a simple truth. Certain plants bloomed only at specific times of the year, almost as if following an invisible calendar. How did a plant know when spring had truly arrived or when the days were shortening for autumn? It was a puzzle that hinted at an internal sense of time. The answer began to emerge in the early 20th century with a fascinating discovery known as photoperiodism, plants sensing the length of day or night. In 1920, W. Garner and H. A. Allard, working with a peculiar tobacco mutant called Maryland mammoth, made a pivotal observation. This tobacco strain grew enormous leaves but refused to flower during the summer in Maryland. It seemed to be waiting for something. They brought the plants into controlled environments, manipulating the hours of light and darkness the plants received each day. To their surprise, it wasn't the day length that mattered most, but the uninterrupted period of darkness. The plants were counting the night. This led to the classification of short-day plants like the Maryland mammoth, which flower when nights are long, and long-day plants, which flower when nights are short. This profound insight suggested plants possessed an internal clock, a sophisticated mechanism to measure time and synchronize their life cycles with the seasons. But how did plants actually see the light and dark? What was the molecular switch that registered these subtle changes in illumination? Decades later, the answer arrived with the discovery of phytochromes, a family of pigments that act as the plant's primary light sensors. Phytochromes are remarkable. They can absorb both red light and far red light, changing their shape and activity depending on the type of light received. They exist in two interconvertible forms, PR, which absorbs red light, and PFR, which absorbs far red light and is generally the biologically active form. During daylight, red light is abundant, converting PR to PFR. At dusk, far red light becomes more prevalent and PFR slowly reverts to PR in the dark. The ratio of PR to PFR acts like a precise switch, signaling the plant about the length of the day and, crucially, the length of the night. This elegant mechanism allows plants to fine-tune their flowering time, ensuring they bloom when conditions are most favorable for reproduction. But light wasn't the only cue. Some plants seem to possess another kind of memory, one tied not to light, but to temperature. This phenomenon, known as vernalization, described how certain plants require a period of cold exposure before they can flower, even after warm conditions return. Consider winter wheat. Planted in autumn, it endures the cold months, seemingly dormant, only to burst into reproductive life the following spring. Without this essential cold period, the plants would simply continue vegetative growth, never producing grain. The cold prevented premature flowering. Scientists began to experiment, exposing seeds or young plants to controlled periods of chilling temperatures before planting them in warmer conditions. The plants remembered the cold. Even weeks or months later, when warmth returned, they proceeded to flower, demonstrating a remarkable thermal memory. How could a plant retain this memory through cell divisions and changes in season? The mechanism remained elusive for decades. The breakthrough came with the advent of molecular biology, allowing scientists to pinpoint specific genes involved in flowering pathways. A key player was identified in the model plant Arabidopsis thaliana*, a gene called *FLC*, or flowering locus C. *FLC* acts as a potent repressor of flowering. When *FLC* is active, it prevents the plant from initiating flower development. The magic of vernalization lies in its ability to silence *FLC*. The prolonged cold gradually switches off this repressor gene. This silencing isn't a change to the DNA sequence itself, but an epigenetic modification. It's like adding a stable bookmark to the gene. Specifically, cold causes changes in chromatin structure around the *FLC* gene, making it inaccessible to the cellular machinery that would normally activate it. This epigenetic mark is remarkably stable. It persists through cell divisions, ensuring that the plant remembers its cold exposure even after temperatures rise. Once *FLC* is silenced, the brakes on flowering are released. The plant is now free to respond to the warmth and longer days of spring, finally blooming. This intricate dance between temperature, genes, and epigenetics reveals a sophisticated mechanism for plants to adapt their life cycles. Such precise timing is crucial for survival, ensuring that reproduction occurs when pollinators are active and resources are abundant. Understanding these memory mechanisms is increasingly vital as climates shift, impacting agricultural yields and wild plant populations. The ability to count day length, sense temperature, and retain this information over time challenges our conventional notions of plant intelligence. These are not mere passive responses but active computations, allowing plants to make complex decisions about their most critical life events. Each discovery, from early observations to molecular breakthroughs, represents countless hours of quiet perseverance in labs and fields. As we reflect on how flowers remember the passage of time, we are reminded of the botanical world's deep, almost human-like capacity for memory. Next, we will journey further into the astonishing intelligence of the botanical world, exploring how plants make decisions and learn. As we reflected on how flowers remember the passage of time, we touched upon a deep, almost human-like capacity for memory. This leads us to an even bolder question: can plants exhibit intelligence, making decisions and learning without anything resembling a brain? For a long time, the very notion of plant intelligence was met with skepticism. How could an organism rooted in place, lacking a nervous system, possibly engage in problem-solving or adaptive behavior? Yet, if we redefine intelligence not by the presence of a brain, but by the ability to sense, process information, and respond adaptively to environmental challenges, the botanical world offers astonishing examples. It's a quiet form of ingenuity expressed through chemical signals, growth patterns, and a profound sensitivity to their surroundings. Plants are constantly making choices, albeit at a slower pace than animals. Consider the climbing vine, a plant that must find support to reach the sunlight. It doesn't have eyes or muscles, yet its tendrils embark on a deliberate search, a silent quest for stability. The tendril, a slender, whip-like extension, doesn't grow randomly. It spirals and sweeps through the air, sensing its surroundings with remarkable precision. When it encounters a surface, a branch, a wall, another plant's specialized cells within the tendril detect the physical contact. This isn't just a brush, it's a recognized signal. Upon contact, the tendril begins to coil tightly around the support. This coiling is not a simple mechanical reaction, it's a directed movement powered by differential growth. One side of the tendril grows faster than the other, causing it to curl and grip. It effectively chooses its anchor, securing its ascent towards the light. This ability to explore, sense, and respond to physical cues in a goal-oriented way demonstrates a form of problem-solving, a silent botanical intelligence at work. The vine navigates its environment, making decisions about where and how to grow, optimizing its chances of survival and reproduction. It's a testament to adaptive behavior, a complex interplay of internal growth regulators and external environmental signals. Even more astonishing is the sensitive plant Mimosa pudica *. Its feathery leaves fold inwards dramatically at the slightest touch, a rapid, visible response. This movement, powered by specialized structures called pulvini at the base of each leaflet, is a defense mechanism against herbivores or strong winds. Early observations considered this a simple, programmed reflex. A touch, a chemical signal, a rapid leaf collapse, predictable and straightforward. However, experiments by Monica Gagliano and colleagues revealed something far more profound. They subjected mimosa plants to repeated, harmless stimuli, like dropping them a short distance. Initially, the plants responded by folding their leaves, but after several repetitions, they stopped. They learned that the stimulus was not a threat and conserved their energy. This phenomenon, known as habituation, is a basic form of learning. The plants showed a form of memory, retaining this learned behavior for weeks, even without further training. It was a startling discovery, challenging the very definition of what constitutes learning. How could a plant, without a brain, exhibit such a sophisticated cognitive process? This led to intense discussions and the emergence of plant neurobiology, a field exploring complex signaling and information processing in plants, often drawing parallels to nervous systems. It's important to clarify, no one suggests plants have literal brains or neurons like animals. The term neurobiology is used metaphorically to describe complex electrical and chemical communication. Plants possess intricate networks of cells that transmit signals over long distances, integrating information from various parts of the organism and its environment. These signals, often involving calcium ions and plant hormones, orchestrate responses from growth adjustments to defense mechanisms, acting as a decentralized command system. The root apex, the very tip of the root, has been proposed by some as a brain-like center, capable of sensing, processing, and directing growth decisions. While the term brain remains highly contentious, the evidence for sophisticated information processing, memory, and decision-making in plants continues to mount. It forces us to broaden our understanding of intelligence itself, moving beyond an animal-centric view to appreciate the diverse ways life can adapt and thrive. This redefinition allows us to see the botanical world not as passive scenery, but as active participants in a complex, intelligent dance of life. Beyond individual problem-solving, plants also demonstrate remarkable intelligence in their communities, much like the forest networks we explored earlier. When a plant is attacked by herbivores or pathogens, it doesn't just suffer in silence. It sends out distress signals, a chemical cry for help. These signals often take the form of volatile organic compounds, VOC, airborne chemicals that carry information through the air. Neighboring plants, even those of different species, can eavesdrop on these chemical warnings. They detect the VOCs and interpret the message. Upon receiving a warning, healthy plants can then prime their defenses. They might start producing protective chemicals, strengthening their cell walls, or releasing their own deterrents. This collective defense strategy, a coordinated response to threat, speaks volumes about the communicative and adaptive capabilities of plant communities. It's a form of distributed intelligence where information is shared and actions are coordinated across an entire botanical landscape without a central command center. From the tendrils' search for support to the mimosa's learned habituation and the forest's coordinated defense, plants demonstrate a profound ability to interact with their world. This journey through their clever strategies, ancient networks, whispering roots, and time-keeping flowers reveals a botanical world far more dynamic and sentient than once imagined. The patience and ingenuity of researchers, from Darwin's careful observations to modern molecular techniques, have slowly peeled back the layers of this green mystery. We are left with a deeper appreciation for the astonishing intelligence that surrounds us, often hidden in plain sight, challenging our very definitions of life and consciousness. The silent decisions of plants, their quiet learning, and their collective wisdom remind us that intelligence takes many forms, each perfectly adapted to its unique existence. As these botanical brains continue their quiet work, processing information and adapting to an ever-changing world, we are left to ponder what other secrets they hold. Next, we will synthesize these revelations, drawing together the astonishing intelligence of the botanical world into a final, reflective chapter. Our quiet journey through the botanical world has brought us to this final reflection. We began by marveling at the sheer ingenuity of carnivorous plants, those silent hunters defying our expectations of what a plant could be. From the snap of the Venus flytrap, discerning touch from rain, to the deceptive allure of pitcher plants, we saw evolution craft astonishing solutions to nutrient-poor environments. Then we delved beneath the soil, discovering the ancient, invisible wood wide web, a vast fungal network connecting entire forest communities in a silent, cooperative embrace. Mother trees, sharing vital resources and warnings, revealed a complex intelligence, challenging our notion of individual struggle in the wild. Further still, we explored the intimate whispers of roots, their chemical language, sensing their environment, recognizing kin, and navigating the subterranean world with deliberate precision. Auxins and other hormones orchestrated their every move, while root exudates formed a constant dialogue with the microscopic life of the rhizosphere, a bustling chemical metropolis. We then turn to the skies, finding that flowers are sophisticated timekeepers, counting the hours of darkness and remembering the chill of winter through the subtle magic of photoperiodism and vernalization. Phytochromes and epigenetic switches revealed how plants measure seasons, ensuring their most vital act, reproduction, is perfectly timed. Finally, we confronted the boldest idea: plant intelligence. From the problem-solving tendrils of a climbing vine to the learned habituation of Mimosa pudica, we saw active decision-making without a brain. These were not mere reflexes, but complex information processing, a distributed intelligence that challenges our very definitions of sentience and awareness. This journey profoundly shifts our perception. Plants are no longer just passive resources, silent backdrops to animal life. They are active, responsive, and incredibly complex beings. For much of human history, plants were seen primarily through the lens of utility: food, shelter, medicine, fuel. Their inner lives were scarcely considered. But the patient work of generations of scientists has slowly unveiled a hidden world, revealing layers of sophistication that demand our attention and respect. This expanded understanding brings with it ethical considerations. If plants can communicate, learn, and even remember, how should we view our interactions with them? Does an awareness of their intricate lives imply a greater responsibility from us beyond simple stewardship? It prompts us to reconsider our place within the living tapestry. It's not about anthropomorphizing plants or claiming they have human-like consciousness. It's about recognizing their unique form of intelligence, perfectly adapted to their existence. This forces us to broaden our definition of intelligence itself, to encompass the myriad ways life processes information and navigates its world. The mystery isn't gone, it has deepened. We realize how much more there is to learn, how much more subtle wisdom is held within the green silence. Every rustling leaf, every unfolding bud, every hidden root system is a testament to an interconnectedness we are only just beginning to truly grasp. This awakening calls for humility, acknowledging that our understanding of life's complexities is still in its infancy, especially concerning our silent green neighbors. Beyond the science, there is the sheer, undeniable beauty. The intricate patterns of a leaf, the perfect symmetry of a flower, the patient strength of a tree. Now, knowing the unseen intricacy beneath, the beauty becomes even more profound, infused with the knowledge of hidden networks and silent communications. This deeper appreciation naturally inspires a profound respect, not just for individual plants, but for the entire botanical kingdom that sustains all life on Earth. Plants are the quiet architects of our planet, shaping the atmosphere, building the soil, and forming the base of nearly every food web. Their vital role, often taken for granted, becomes impossible to overlook once their active, intelligent engagement with the world is revealed. This understanding fuels the urgency of conservation. Protecting plant diversity is not just about saving species. It's about preserving entire libraries of wisdom and ingenuity. Each plant species holds unique adaptations, unique solutions to life's challenges, a testament to millions of years of evolutionary experimentation. As our climate changes, understanding these botanical intelligences becomes ever more critical for predicting and mitigating environmental shifts. A future where humanity thrives in harmony with nature depends on our ability to listen to the silent wisdom of plants, to understand their needs, and to protect their capacity to adapt. It calls for a partnership, a recognition that we are not separate from the green world, but intimately woven into its fabric, dependent on its silent, ceaseless work. So, as our story draws to a close, let this be a call to renewed curiosity. Look at the plants around you with fresh eyes, with a deeper appreciation. The tree outside your window, the flower in your garden, the grass beneath your feet, each holds a universe of complexity, a quiet intelligence unfolding. Listen to the silence and you might hear the whispers of roots, the ancient hum of forest networks, the patient count of a flower. Science, in its pursuit of knowledge, often leads us to greater humility, revealing the vastness of what we do not yet comprehend. The mystery isn't solved, it's simply opened wider, inviting us to keep exploring, to keep asking questions, to keep learning from the green world. Our relationship with plants is a continuous dialogue, a conversation that grows richer with every new discovery, every quiet observation. They teach us about resilience, about adaptation, about finding ingenious solutions to life's most fundamental challenges, often without fanfare or grand gestures. They are the quiet giants, the silent partners, the foundational wisdom of our living planet. Even now, as the world slows for the night, countless botanical processes continue, unseen, unheard, shaping the very air we breathe, the soil beneath our feet. And so, as the moon climbs high, casting long shadows over the quiet, intelligent green world, may you sleep well, filled with wonder for our astonishing plant neighbors.